next speaker is Masataka Kawamura, and his presentation is entitled Normothermic Ex-vivo Kidney Perfusion Preserves ATP Generation and Graft Function After Warm Ischemia, which is further enhanced with pharmacologic mitochondrial protection. Dr. Kawamura graduated from Nagoya City Medical School in 2011 and trained as a junior resident before joining the Department of Urology at Osaka University, where he trained as a senior resident. During this time, he received training in kidney transplantation, urological oncology, and stones, and obtained the Urological Society of Japan specialty certification. He has also worked at Osaka University Hospital, one of Japan's leading renal transplantation centers. And in 2017, he began research on reducing ischemia reperfusion injury in renal transplantation as a graduate student at the Osaka University Graduate School of Medicine, where he received his PhD in March 2021. With that, we'll welcome Dr. Kawamura.
Nice to meet you, everyone. It's a great honor to give a talk today. I'm going to present our latest research work about Normothermic Ex-vivo kidney machine perfusion. This is our warm perfusion system. It can maintain organs at physiological temperatures, arterial pressures, oxygen, and carbon dioxide in the perfusate. And we use erythrocyte-based oxygen carrier as a perfusate. And during perfusion, there's no cold ischemic injury. And there's also a possibility to assess graft function. And there's also a possibility to offer new treatment to modify organ damage. This is a previous study about transcriptome analysis. In this study, mitochondrial metabolism genes are enriched in warm perfusion group compared with a static cold storage group. And this proteomics analysis also showed warm perfusion preserved expression of key mitochondrial proteins. From these studies, we hypothesized that warm perfusion could maintain cell metabolism and mitochondrial function during organ preservation. To prove this hypothesis, we conducted this animal experimental protocol. After 60-minute warm ischemia, kidneys were taken out from 30-kilogram pigs, and then kidneys were subjected to 5 hours of cold storage or warm perfusion. And then we performed autotransplant with contralateral nephrectomy. And then pigs were followed for 3 days. In no warm ischemia group, immediately after taking out the kidneys, we performed autotransplant. We first assessed the graft function after implantation. As you can see, in cold storage group, serum creatinine levels went up to 12 at POD3, while in warm perfusion group, this increase was significantly suppressed. And the result of BUN was consistent with the result of creatinine. And then creatinine clearance and fractional excretion of sodium, or FeNA, at POD3 were also calculated using 24-hour storage sample. And as you can see in the warm perfusion group, FeNA was significantly lower than cold storage group. And then we assessed ATP levels in the live renal cells. First, we extracted the cells from the renal tissue taken by needle biopsy gun, and then mix the cells with this reagent, and then luminescence was measured. This is the result. ATP levels in the renal cells were significantly preserved in warm perfusion group compared with cold storage group at both time of after storage and 30 minutes post reperfusion. Interestingly, the level of the warm perfusion group was almost similar to the level of healthy kidney levels. And we also checked the energetic metabolism to assess mitochondrial function. Seahorse XFE analyzer can measure oxygen consumption rate in live cells which indicates the activity of the mitochondrial respiration. When you look at these curves, in cold storage group, mitochondrial respiration was maintained extremely low, but in contrast, in warm perfusion group, the mitochondrial respiration was well preserved. This was again comparable with healthy kidney level. This is a possible subsequent reaction following ischemia reperfusion in kidneys. Mitochondrial dysfunction would cause release of reactive oxygen species and oxidative stress, followed by the activation of inflammation. Then T-bird is one of the oxidative stress marker for lipid peroxidation. We compared T-birds in the plasma. At POD 2 and 3, we could detect significant difference between cold storage and warm perfusion group. In summary, so far, we demonstrated that warm perfusion preserved ATP levels and cell metabolism in the renal cells during organ preservation. And maintenance of mitochondrial function might be crucial in organ preservation. Here is our basic Perfusate composition. We next hypothesized that supplementation of mitochondria-targeted drug would further improve cell metabolism and graft function. AP39 is the drug we decided to use. It consists of mitochondria-targeted motif and hydrogen sulfide donating moiety. This is a possible mechanism of action by AP39. Hydrogen sulfide released from AP39 could serve as an electron donor. In mitochondrial membrane, AP39 supports electron transport and the generation of ATP level. In this second animal experimental protocol, we compared warm perfusion with and without AP39. The time of ischemic time and time of perfusion was the same as the first protocol. First, some perfusion parameters were compared between two groups. These two parameters were reported to be correlated with the graft function. AP39 group had a significantly better renal blood flow rate and intravascular resistance at 5 hours. Total amount of urine output was also improved by adding AP39 in the Perfusate. By the way, in this study, renal blood flow rate and total urine output during warm perfusion were well correlated with the creatinine level at POD3. And we assumed that especially total urine output could be a strong indicator for the graft function after implantation. And then graft function after implantation was compared between two groups. Serum creatinine and FeNA at POD3 were all, both improved by the AP39. And ATP levels in the renal cells extracted from the cortex tissue was also enhanced by the AP39. And when you look at the result of the seahorse analysis, AP39 could further enhance the mitochondrial respiration. And subsequently, the oxidative stress marker was compared. We measured T-birds in the Perfusate and plasma. Especially in the Perfusate, AP39 could improve the oxidative stress even from the early time point, like 1 hour. In summary, for this study, supplementation of AP39 mitochondria-targeted hydrogen sulfide donor enhances mitochondrial function and reduces graft injury. This could be a candidate for the future treatment to modify damaged organ. This is my final slide. I'd like to thank my PIs, Marcus Seltner and Lisa Robinson, and my colleagues. Thank you so much.
Do we have any questions from the online, the virtual? Nope. Any questions from the audience? Don't be shy.
Very nice work. I was wondering, did you look at the kidneys after in terms of pathology, cortex, medulla, and how does it affect your AP31 treatment on injury?
We checked the pulse staining, and we scored the tubular injury score. We couldn't see significant difference, but there's some trend. For like AP39 could reduce the ischemia, tubular injury score. Yeah. And we are now trying to visualize mitochondria as well. Yeah.
Any more questions? I do have a question. What are your thoughts on why adding a AP39 and how it decreased vascular resistance? Do you have any reason behind that?
I'm not so sure the exact reason, but we put the AP39 also in the UW solution before putting the organ on, and that might be a kind of pre-treatment, and then maybe, and then so we can preserve the ATP levels to some extent after starting the warm perfusion. That will be kind of reperfusion. So maybe this pre-treatment could have some effect on the vascular resistance. That's my thought.
If there are no other questions, we'll move on to our final speaker. That was wonderful. Thank you.